Hello and welcome to Beyond Biotech podcast number 24 and the last one in November. And I've still not decided when to give the podcast a bit of a rest for the holidays. I'm Jim Cornell from the Biotech, and one thing I do know is that yesterday was Thanksgiving in the US, so that meant no emails or press releases from that side of the Atlantic for a couple of days. Maybe you're listening to this in the middle of a big Black Friday line somewhere, in which case I hope you grabbed a bargain. I really enjoyed the planning part of Black Friday, sifting through all the flyers and deciding where to go. The stores here in the UK have taken Black Friday on and they've tried to run with it, although here it can be a little underwhelming. You'll see stores doing something like buy one, get one at 5% off, which to me isn't exactly something worth camping out in front of a store for. One thing I could have used is a good deal on a music system, as mine died this week. The CD player doesn't work anymore. It was old, but still it's annoying. We're in the middle of a lot of strikes here too, so today there's no school, no mail, and there are also train strikes happening quite often. Tomorrow I will be on a train, only it will be in Germany as I make my way from home to Strasbourg for next week's BioFit. There's still time to set up an interview if you're going to the event through the BioFit partnering platform. I'll also be moderating a session based on finding talent. So clearly the reasoning behind that one was, let's find a presenter without talent to exemplify the problem. But it's going to be good to meet some new people and who knows, maybe even see some familiar faces as well. Speaking of events, 3 of the guests this week are from recent events that I've attended, but more on the exact details when I do the introductions. We have interviews with Franjo Hanzl, Vice President Commercial Development Europe at Eversana, Stacy Blain, Founder and CEO of Concarlo Therapeutics, Carl Borrebaeck, Chairman of the Board of Directors of Immunovia and Professor at the Department of Immunotechnology at the University of Lund in Sweden, as well as Jens Hennecke, Vice President of Business Development at SOTIO Biotech. And this week, because of Thanksgiving, there's no chat with Travis McCready over at JLL. Now it's time for the news you may have missed over at Labiotech.eu. And as I mentioned earlier, not a lot coming out of the US for the most part this week because of Thanksgiving. We had an article on FMRP, the protein behind immunotherapy resistance. The Genomic Data Infrastructure Ireland project has been launched to support the discovery of genetic causes of disease, and an agreement has been signed to boost mRNA manufacturing in Singapore. New replay HSV gene therapy company Telaria has been launched to focus on skin diseases. Imorna has been cleared for an mRNA-based COVID vaccine study, and Legend Biotech's small cell lung cancer drug has received FDA approval for clinical development. Fog Pharma raised $178 million to target cancer. Arecor is starting its second type 2 diabetes clinical trial, and Achilles Therapeutics received a US patent for immunotherapies. The FDA approved CSL's gene therapy for haemophilia B. Novo Nordisk announced a $750 million facility expansion, and Chiesi marked the start of a biotech centre of excellence for the future of medicine in Parma in Italy. Immune cells in ALS patients can predict the course of the disease. A trial is about to start for a new heart attack drug, and funding has been announced to develop a wearable patch for type 2 diabetes. Chondrion's mitochondrial diseases treatment is producing positive results. A study expressed concern that the newest COVID subvariant is resistant to all therapeutic antibodies. And we had an article on what recent mixed clinical results can teach about Alzheimer's disease. BIA and ExoPharm have signed a joint research agreement to develop integrated technology. Gene therapy approval is a game changer in haemophilia B treatment, and you can read all of these and lots more at Labiotech.eu. One thing I didn't mention there is that this week we launched a new page, and it's a new resource, which is a listing of all of the different science and health awareness days, weeks and months. And it's quite a long list, especially May. Anyway, let's get to the interviews, and there are 4 this week. It's always a challenge deciding on the order, but today we'll kick off with Jens Hennecke, Vice President of Business Development at SOTIO Biotech, who I had the pleasure of chatting with at Bio Europe recently. And rather than telling you about SOTIO Biotech, I'll let Jens do it. And I guess the easiest question is to tell me about the company.
Absolutely, happy to do that. So at SOTIO we are developing oncology products, so basically a pipeline of different classes of oncology products, all for the treatment of solid tumors. So we are not so much focused on hematologic cancers, but really on solid tumors. And have 3 classes of products. One is cytokine-based, IL-15-based products, different types of products, we can talk about them. We also develop antibody-drug conjugates, as well as T-cell products. So it's a very diverse activity of different products, different classes of products. We're always on each platform. We have a lead program already in phase 1 or phase 2 clinical trials, and then a pipeline on each of the platforms that follows thereafter.
And do they work in combination with each other, or are they all separate trials?
So we have set it up, and these basically are all products or platforms that we brought in through different types of business development activities. So these are licensing, collaborations, etc. They all have a standalone rationale, and they also have a independent risk profile. So they are not. It's not that one product relies or one platform relies on the rationale and the success of the other program. So they all, it's basically the risk mitigating approach for building a broader pipeline of products. There is some synergies indeed. One synergy that we are exploring is definitely the one which is obvious between the IL-15 platform as well as the T-cell therapies.
Right. Could you tell me a little bit about each of them and how they work?
Absolutely. So let's start with what we consider our lead program, biggest value driver in the company, which is our IL-15 program called SOT101. What we did there is we took IL-15 and engineered it so that it has a very unique specificity with regard to the targeting receptor. It's designed to activate T cells and NK cells that have the so-called IL-15 beta-gamma receptor on the cell surface, and do that independently of antigen-presenting cells. So meaning designed so that the molecule also activates T cells and NK cells in the tumor microenvironment, which is where this other receptor type is not available. We develop a program there already for 3 years in the clinic. We have completed phase 1 as a monotherapy dose escalation study that has shown very nice single agent activity. We also have done a phase 1 dose escalation in combination with pembrolizumab, which has shown very nice response, especially also in patients that have been checkpoint inhibitor pre-treated and have failed for prior checkpoint inhibitor treatment. And this is a program that we have now advanced to phase 2 clinical studies. So it's really the rationale of activating T cells and NK cells to enhance the effect that the other drugs also provide. So in this case, we are we are running a broad phase 2 clinical study in combination with pembrolizumab. It's a 300 patient study in phase 2, testing the combination in 6 indications. And that is really currently the key activity. We're also running monotherapy studies. We are also about to start another combination with cetuximab, so always relying on the activation of T cells and NK cells. And based on this modality of the IL-15, we also develop targeted cytokines, so targeted IL-15s, either in terms of tumor targeting, so the classical immunocytokines, where you fuse this engineered cytokine to a tumor targeting antibody, or where you actually combine it or fuse it to also to checkpoint inhibitors. So these are earlier stage activities that where we build kind of the next part of the pipeline upon. The second aspect of the pipeline is a very big activity, our antibody-drug conjugates. This is relying on 2 deals that we have done, one with Basel-based NBE Therapeutics that is now part of Boehringer Ingelheim, and a second deal that we have with LegoChem from South Korea. 2 very beautiful platforms that deliver very well-defined and stable antibody-drug conjugates with also unique toxins. And here the lead program on the platform is based on the NBE technology that has now started phase 1 clinical trials. So that's a program targeting an antigen called Claudin 18.2 for the treatment of gastric and pancreatic cancer. And that's in the middle of dose escalation study. We don't have data there yet, but the distinguishing factor here is on the one side the platform from NBE with a very unique anthracycline-based toxin that so far hasn't been tested in clinical trials, and on the other side having generated an antibody that seems to be superior to all other Claudin 18.2 antibodies that we have seen out there in the industry. And then the third pillar is our so-called Boxer platform. That's our T-cell therapy platform. That one we acquired about 2 years ago from a company, Unum Therapeutics from Boston. The idea here behind this platform is to enhance the metabolic fitness of T cells in the tumor microenvironment. So the idea is that you co-express certain genes together with, in this case, the CAR T, or the CAR, in the T cells, and you can then show in nicely in animal models that the persistence of the cell is increased, the cells are active in hypoxic conditions, in low glucose conditions, basically the conditions that you typically find in a solid tumor microenvironment. And that's a concept now also we're going to test in the clinic. So the program is about to start phase 1. So it's just waiting to include the first patient.
Right. And the timelines for those three, where are you at with all of the three in terms of being able to bring these to market?
So market, I would say, is a bit out, right? I mean, here we're looking more towards the kind of mid-end 20s, really, right? For us right now, especially the next 1.5 years are exciting to generate a clinical proof of concept. For the IL-15 program, we believe we already have it, but need to kind of solidify that or make that statistically, that effect that we see kind of prove that within a more statistically significant manner. This trial, basically all these trials, whether it's phase 1 trials or phase 2 trials, they all have readouts between 1 year and 2 years. So really the next 48 months are very exciting for us and will be pivotal for the company, and will show kind of which of those hypotheses work out. And then always with the earlier pipeline, we can then accelerate the activities on that respective platform.
And so what brings you to BioEurope or partnering opportunities?
Let's say the last couple of years we have always looked for new platforms. That one we have tuned down a bit. Right now the name of the game is making sure we deliver on our own programs, right? And it's the typical activities with, are there partnerships possible at this point in time? What data does a partner need? So we're always looking for potential partnerships on the programs, especially the clinical programs. The other aspect is, of course, it's also an investor reach out, because we have in mind, or may have in mind, to take third-party investors on board. Right now, SOTIO is fully funded by one single investor, and that one we are planning to change over the next 1 to 2 years.
Next, it's to another event I attended, and that's the most recent one, the Medicon Valley Alliance Annual Summit, which took place in Copenhagen at the beginning of this month. I have a few videos to edit from all of the events, but this was an audio-only one with one of the Alliance members, Eversana, and I had a conversation with Franjo Hanzl, Vice President of Commercial Development Europe at the company, while they were clearing away lunch, so apologies for the background noise of lots of plates banging together. Obvious first question is if you could tell me a little bit about the company that you're with.
Sure. So I work for Eversana. Eversana is American-based venture capital-owned company which provides services to the pharma industry. We are 7,000 employees, just over 1 billion in revenues, and we are serving more than 600 clients all over the globe, from big pharma, med tech, oncology companies, but also then first-time launchers. And that's why we are here, right? To help the companies from Medicon Valley Alliance that are first time looking into their assets, how they're going to commercialize them, to better understand their market opportunity, better understand the competitive landscape, better understand the profile of the markets, sequencing where they should launch first globally. Is it US or Europe or vice versa or somewhere else? And then when they figure out how they're going to do it, whether they want to do it by themselves or they want to do it out licensing, or they are maybe thinking about by themselves but using and relying on external support through outsourcing of the entire commercial organization. This is then where we step in and we actually set up and execute the entire global commercial infrastructure on their behalf to allow them to launch the products to the market, but keep the intellectual property with themselves, which increase their stock price if they are IPO or increases their market evaluation as the company, and not necessarily out-licensing their assets to someone else.
And I guess it varies company to company what their actual needs are.
Obviously, here we are talking about 300 members at Medicon Valley Alliance, another couple hundred with Medicon Valley Lund, another couple hundred with Oslo Cancer Cluster. So Nordics, we have seen this morning, are tremendous opportunity in terms of the number of the biotech companies, medtech companies, digital medicine companies that are looking into solution, how they're going to bring their products to the market. Right?
And I guess that when people start companies for startups, they're really concentrating on the science and don't really have any grasp of the kind of things that you can offer.
That's correct. So the companies are focusing on science, on meeting the regulatory agencies' needs, like European Medical Agency or FDA and the MHRA in UK, etc. But actually, that's not what makes the product successful to the market. You also need to have a perspective on the value that you're creating, whether this product is competitive, how the stakeholders like payers, reimbursement, hospital, formularies will actually look at the value of the product. So it's not only satisfying the regulatory agencies' demands, it's really satisfying payers, but also providers like physicians, patients, whether there's an unmet need that many different stakeholders will need their product, right? So this is all where Eversana can help through our management consulting agency, which is called Eversana Management Consulting, which is part of our services next to channel, distribution, data analytics, patient support services, and other services that are needed as virtual pharma company.
And how do you get the word out to those companies that you exist?
We are a member of Medicon Valley Alliance, so there are a number of opportunities in their internal meetings or sponsored meetings where we bring in experts. We talk about oncology, we talk about regulatory, we talk about payers, landscape, we talk about specific topics that are of an interest to the members of Medicon Valley Alliance, and then we spread the word of what we do. So it's been since let's say over the last 12 months we have been more present here when we became a member. So we are in our first year of membership. I believe we had around 40 different company discussions in how Eversana can help them. So there is certainly a need and gap in the market. We are the only company of this kind in the world.
And do you deal with other areas as well, or just in this region?
Only life sciences. So we are global, right? So we are 7,000 employees. We have 20 offices in US. We have 10 different offices in Europe, in London, Berlin, in other cities. We have a full presence in Japan, Asia Pacific. So we serve life sciences all around the globe.
So you can help anyone?
Almost everyone.
Next, it's a trip back to the Medicon Village, which is in Lund in Sweden. I took some time out from NLS days while I was in Malmö to visit the amazing Medicon Village site. But while I was there, I interviewed someone who could be described as a giant of biotech, Carl Borrebaeck. If I read out his biography, it would be longer than the interview, but suffice it to say he's been involved with lots of companies, hence the connection to Medicon Village, and he's chairman of the board of directors of Immunovia, and he is also professor at the Department of Immunotechnology at the University of Lund in Sweden, as well as being director of Create Health. He's the former vice president of the University of Lund and received the Nobel Science Award 2009 for contributions to cancer proteomics and antibody-based therapy, and lots more awards as well. But in spite of all that, he's a very warm and humble person to speak to, so here's our conversation. Yeah, so I guess if you could tell me a little about yourself and your involvement in life sciences.
I mean, the background is that I'm a professor of immunology, cancer immunology. I've been working in cancer area for 20 plus years, and always thought it would be very rewarding to see if we can get some of the research result out into the society. I mean, there are studies showing that it takes 17 years for the discovery before it goes into patients. So we started a translational cancer center 15 years ago. It's still alive. Create Health is the name of it. And we were around 70 people, 7 PIs. I was the director. And the vision we had was we do this for today's generation and not next generation, to get it out quickly. So we actually managed to get some of the research results out, both as therapeutics into clinical trials and also in diagnostics. But I quickly realized that it's not really in the academic mission to do this. We have to translate it into a commercial entity. So we started a company. The first company I started was probably 25 years ago, BioInvent, which was a cancer therapeutic company. It was followed by Alligator Bioscience, also in immuno-oncology, one of the Nobel Prizes some years ago. And then we started Immunovia, which is a cancer diagnostic company, now going into the US market as the first ever having a test for pancreatic cancer, early diagnosis of pancreatic cancer. Pancreatic cancer, 5-year survival is about 4 or 5%. It's really a horrible disease. But if you find it early with our test, you can increase the 5-year survival up to 50%, 55% even. So it's a tremendous gain. And coming from academia, then slowly going into more and more commercial activities. I'm still a professor at the university, but find it very rewarding for both me and society, and the students are really excited about seeing what they've done, every night in months and months, actually become application. Another area we worked in was to understand how the immune system reacts, not only against cancer cells, but also allergens. We have a tremendous increase in hypersensitivity in the society. So based on that, we started another company, Sensagen. They're all here in the Medicon Village. Sensagen is now producing a test that can determine if a chemical is sensitizer or if it's not a sensitizer. That has a tremendous application in cosmetics, in food, medical device, almost anything. The color of your shirt has been tested. The rubber on my boots has been tested. And that company goes very well and just got an OECD approval. And then the latest, I wouldn't say the last company, but the latest company is the digital tool for self-management of chronic pain. So it's completely different from the other one, both in the area, but also in the indication. About 20 to 25% of the general population suffer from chronic pain. That's 190 million in the US and EU. And very few, 2-3%, actually come to our pain specialists because they are rare, difficult to find, and it's costly. So we developed a digital tool. It's an application. You have it in your telephone, where you can understand what actually gives you pain. The activity and everything you do during the day results in pain. And we're just finishing 3 clinical studies in the US, San Diego, Boston, and New York, and we have just fantastic data out of that. So that's now going into a commercial phase in Q1. The other companies are already commercial. I mean, they're selling products, they are establishing themselves in the US, like Immunovia. Sensagen is selling all over the world. And to have them here in Medical Village has been a privilege, because the university, when I was the vice president of Lund University, in charge of innovation and commercialization. So we moved here, most of the cancer scientists, 2012, I think it was. And then have the companies here. It's really, I call it the perfect storm once. You have everything here. It's very easy, also for new startups here at the Medical Village, and it's a unique environment. You don't find anything like this in Scandinavia. I could maybe point you out, there's some in Italy and Texas maybe, but I mean it's so unique. You have basic science, translational science, you have small startups, you have medium-sized companies, you have patent offices, you have business advice, you pretty much have what you need to start a company. When I started the first company, we didn't have anything. We sort of struggled along and tried to find the best investor, and now you can lift the phone and you find investors or patent attorneys or whatever in a minute. So it's a big change. But I think the main thing for this, it's a breeding place for innovations. It's so dynamic, but it's really a nice place to work. And in the summer, not now because now it's horrible weather, it's a really nice campus, because it used to be AstraZeneca's site, and they built it like a big meeting place. So there are fountains, there are sofas, there are everything. You can just walk out and have a meeting with someone. If you go to the restaurant, you meet pretty much everyone you want to meet, from investors to business advice to whatever. So it's quite unique to be here, and we didn't have that when we started the first company or the second company, not even the third company. But now Sensagen and Pain Drainer is the last company, have really benefited from being here. So that's a quick resume.
Sure. Is there a lot of, you mentioned there's a lot of companies here, is there a lot of communication between them?
I mean, a number of companies were spun out of Astra, but those are most service companies, and they live and thrive on the other companies because they sell their services, from pharma development, from manufacturing to reimbursement questions for patent issues and so on and so forth. But also between the other companies, I would say there's a fair exchange of ideas, of experience. I mean, as you know, the biggest problem for all startup companies is money. So there is also an investor community here where you exchange ideas and help each other. Yeah. So I think that's also, I remember the first time we moved into Ideon, which was supposed to be a science park, but it turned out to be more like a sort of researchers' hotel. So everybody had different apartments, but we never spoke to each other basically, because there was no meeting ground. Here, I mean, as you see, you're coming in and out. I mean, you can't avoid meeting people. So yes, I would say there's an unusual amount of exchange.
It's interesting that you mention all of the different services involved, because I think there are quite a lot of startups. You mentioned money being an issue, but I think that quite often it's the other things that they're not necessarily aware that they're going to have to get into when they start a business.
Yes, absolutely. And there is a mentor program here at the Norme. It's Novo Nordisk who started a mentor program, which is quite unique. And I'm the founding mentor in southern Sweden. And you have to apply to this, and every time you meet a company, there are 3 senior mentors. So it's quite interesting to being a mentor, because you meet other experienced people. And it's surprising still at this day of my life, is that you listen to a company and you start at the end, you say, so what's your product? What you actually selling? Oh no, we're not selling yet. We're developing. And so these types of mentoring sessions are super useful for the companies. Some of them are much more mature and know exactly what they want to do, but they still need help with pitching, with investor relation, investor contacts, and so on and so forth.
So how many companies are you involved with, either on a personal level or on a...
Well, I started myself 5 companies, and 3 of them are still very active. I'm the chairman, so I better be active. I was on the board of a few others in Denmark and so on. But being chairman, it takes a lot of time. So now I'm trying to reduce the number of companies. But all in all, I don't know. I've been in 20, 30 companies on the board, but started 5. So, but I think it's really, and also from the department, the academic department, several of the people who worked there and developed an idea together with me are now working in the companies. So they follow from the idea into a commercial setting. It's a tremendous experience for them, a journey that you rarely see. You always get the question, so how much money have you made? I mean, money is not the issue. It sounds, everybody says that money doesn't matter. Of course money matters, but you don't work all day, all night for 3 weeks because you think you in 10 years going to make some money. You actually do this because it fulfills you to see that your idea, your way of thinking actually turn out to be something that could help mankind. It sounds pretentious. We work in cancer research, and a student asked me, but what you do now will never reach the society, will it? I mean, it takes years and years. So I said, if we can improve the life of one person, at least we've done some good. I've been part of also the financial sector, looking at companies and so on and so forth. And I'd rather be on this side, trying to make something.
The people that you mentioned that were in academia but moved into business, do they stay involved with academia? Because if everybody moves over, then there's a vacuum in academia, I guess.
Both, I say. I mean, some moved forever. Some have one leg in each. And some stay and say, just, this is not for me. I wish you all the best. But, I made quite a few millionaires, I tell you. And that's also a lot of fun to see that, all the hard work that they've done actually paid out. I don't know how many times journalists say, call me when you have a breakthrough. I say, we have breakthroughs almost every week. No, I mean a big breakthrough, when you cured cancer. I say, come on, you got to be kidding me. I call you in 20 years then. Because it's such a diverse disease. But we take small steps maybe not every week, but every month or so, which is sort of what we feed off, the small improvements. We see that we are going in the right direction. And suddenly you have something that can go even further because you can package it as a therapeutic program for a patient or a diagnostic test or what have you. I think just when I was the vice president, the only thing I preached was you have to be aware. To me, there is nothing like basic science and applied science. Today it's so interwoven, and you can study the eye of a fly and suddenly have a new idea of a telescope. You can study what we've done, the pathways of a cell, and suddenly you find a molecule. Oh, maybe if we do like this and this and this could, change the course of a disease. So that's a lot of fun.
Definitely. I always think, I have a 10-year-old son, and I always think of the amount that he has to learn compared to the amount that I had to learn in university. It's basic compared to the science of today.
But both yes and no, because if you go into graduate school today, you could in 4 years do so much more than I could, because at some point you have to manufacture your own equipment. Sometimes you have to now you just buy a kit, and the scientific question you have can be answered in a couple of days. And then that leads to another question, and then another question, another question. So the years goes by, but you can do so much more. I remember I was at a conference in Australia a long time ago, and someone has sequenced, I think it was insulin, and it had taken them, forever, 4 or 5 years. And then at the end of the session, this new technology came along, sequencing technology, and a guy showed how he sequenced a number of proteins in a couple of months. So it's really a paradigm shift. I still remember the look on the face of the guy who's been working for 4 years with one. So the technology is really a driving force for, I mean, when monoclonal antibody came in MRC, Cambridge, their patent office said that there was a limited interest in filing patents or something like that. If anything has changed life science, it's been monoclonal antibodies. I mean, it's a tremendous change. And Greg Winter is a good friend of mine who got the Nobel Prize for phage display, which is the next sort of step in antibody manufacturing. And now, biopharmaceuticals is probably the biggest money maker in pharma industry. So be aware of the next possibility around you.
I guess a lot of people, it's more about the science than thinking in a lot of businesses it's more a case of how can I find a niche, whereas in science it seems to be, well, hopefully more driven by the discoveries and the hope of...
Yeah, but they go hand in hand. Suddenly you discover something. I mean, I say to my B. F. Fleming, when he saw something was not growing in an area and he discovered penicillin, be aware that an experiment that goes wrong might be the best experiments you ever made, because it opened up a new door to something else.
Now an interview that isn't related to an event, although there is mention of one in there, because we're headed to Brooklyn, New York for an enlightening conversation with Stacy Blain, founder and CEO of Concarlo Therapeutics, which is doing some great things in the oncology space.
Concarlo Therapeutics is a preclinical oncology company, and we are focused on changing the outcomes for drug-resistant cancer patients. So we are focused entirely on a novel transformative target called P27, and P27 has the unique ability that it is the natural inhibitor, the way every cell in your body normally regulates the three main drivers of proliferation: CDK4, CDK6, and CDK2. And cancer, as its essence, is a disease of unregulated proliferation. It's a cell that doesn't listen to the signals that tell it to stop dividing or tell it when it should be dividing, and it just divides all the time. And that uncontrolled proliferation is regulated by CDK4, 6, and 2. So there's lots of interest in the oncology space to target and drug those three high-value targets. And we're sort of following nature's lead, and we're drugging P27, getting P27 to do its job and inhibit these three kinases. And we have lots of optionality across numerous tumor types, but one of our initial focuses is on metastatic drug-resistant breast cancer, which is a very large market space. In the US alone, that's about 43,000 women who become metastatic and drug-resistant annually. We have both large biologic assets in our pipeline as well as small chemistry assets in our pipeline. So multiple shots on goal. And we're the first company to drug P27, and we're very excited to bring, first-in-class P27 inhibitors to the market.
So could you tell me a bit about the therapeutic peptides, what they are and how they work?
Sure. So if P27 is the natural inhibitor of these three high-value kinases, so these are the kinases that get dysregulated in every type of cancer and push these cancer cells forward, P27 is the way that normally we stop that. And so think about P27 as a protein that binds to these three targets, CDK4, 6, and 2, and it's an on-off switch. It tells these three kinases, CDK4, 6, and 2, whether they should be on, pushing the cell into grow, or whether those kinases should be off. So it really is turning them on, turning them off. But the simplest way to think about P27 is imagine a door that is a door to a CDK. And so when the door gets modified, the door swings open and the CDK is on. It can push the cell forward. But when the door is not modified, the door slams shut and the CDK is off. So we're designing a series of proteins, of inhibitors, to slam that door shut and block that modification. And we again took our lead from nature because nature has a small protein called ALT that naturally does that. Alt will bind to P27 and block that modification and slam the door shut. So we took Alt and we bioengineered it to make it much smaller so that it could be produced in the synthetic setting, make it drug-like, so to speak. We changed some of the contents of that peptide to make it more stable so it could have extended half-life, right? So that it could actually be used as a drug to get to patients. And then we packaged it in a liposome. And the most famous liposomes, many people in the world now have liposomes in their arms. The COVID vaccines were a piece of genetic material that was packaged in a liposome, or think of a liposome as a lipid suitcase. So we put our therapeutic peptide in the lipid suitcase so that it can protect it when it's circulating in the patient's bloodstream. So the lipid suitcase can help get this therapeutic peptide to the cell, enable it for the peptide to get into the cell. And then when the therapeutic peptide is in the cell, it finds its target P27, it locks P27 into the off conformation and turns CDK4, 6, and 2 off. So we're classified as what's called a biologic. It's based on a peptide, so it has lots of specificity for its target. Think of sort of like zipper-like function. And the benefit of that is it has very little chance of having what's called off-target effects, binding to other things in a normal cell, because that's what is frequently the cause of toxicity. The oncology drug that we give the patient, yes, maybe it hits its target of choice, but it also hits many other targets in the patient's body, and that's what makes the patient sick. So because of the high specificity due to the biology, we think that we can produce a therapy that has really strong differentiators. It is following nature's lead. It is high specificity, low toxicity. And then the other really important thing about this therapeutic peptide is when you use this peptide to inhibit CDK4, 6, and 2 simultaneously in concert, it drives tumor cell killing. So you get tumor regression. You cause the tumor to shrink, as opposed to many other therapies in the oncology space just cause the tumor to stop growing. That's called cytostasis. The problem with cytostasis is we really want to reduce tumors so we can actually extend the overall survival of our patients. So we have a very differentiated mechanism of action. We're using a peptide, which is, a pretty novel approach, and we have a first-in-class target. So very excited to bring this really transformative technology to our patients who are very much in need.
For sure. Is it something that works in tandem with other treatments, or does it work on its own?
That's a great question. So it does work on its own, but at least at this stage, in terms of the frequency and the dose that we're giving to our experimental lab animals, we do get some increased responses in the presence of other drugs. So in the metastatic breast cancer space, there's a very big drug called Ibrance. That is a CDK4 monotherapy inhibitor. So it just inhibits CDK4, 6, not CDK2. That's actually the drug that most of the metastatic breast cancer patients first go on. That's the standard of care therapy. That's a great drug. It was came to market in 2015. It's called Ibrance. There's also another one, Verzenio and Kisqali. Those are really amazing drugs. They essentially keep patients in remission for longer. The problem, however, is that we know with time, essentially every patient is going to become resistant to Ibrance, and they become resistant because CDK2 gets turned on. But in terms of our therapy, we can use it as a monotherapy. Our therapy is called IPY, so we can use it to inhibit CDK4, 6, and 2. But we do get increased synergy when we add it in combination with Ibrance. We get even better responses in our metastatic drug-resistant breast cancer mouse model. So we are exploring combination with CDK4 inhibitors and then other inhibitors, particularly as we are contemplating and moving into different tumor types such as ovarian, endometrial, and uterine, and even big cancer types such as like non-small cell lung cancer, which while we have lots of great first-line therapies, there's still well over 100,000 drug-resistant patients in the US alone on an annual basis. So lots of need in terms of patient numbers and lots of market space in terms of, our company.
And how is the drug administered?
The drug is given by infusion. So currently we are trying to get down to once weekly dosing. We're about at 2 times a week dosing, but we're very cognizant of the patient experience, right? We don't want these metastatic patients to have to be spending a lot of time in the clinic. And we're really working very hard to increase both the time that the drug circulates in the patient and also package more bang per punch, so to speak, like load more of the drug into that lipid suitcase, so that we can get less frequent dosing for our patients. But it is given in a hospital setting, in a transfusion setting.
Is it something that is stopping the cancer from progressing and therefore simply prolonging life, or is it reversing it and getting rid of the cancer?
That's a great question. And I can only answer at this point about what we see in our animals because we haven't yet tested our drug in humans. That's our goal. We're hoping to be first in human in about 15 to 18 months, have our first clinical trial. But what we see in mice is we actually can cause tumor regression. We can take a very large tumor, a tumor that's maybe 700 millimeters cubed in a mouse. And so that's about the size of a quarter or, a little bit bigger than a euro, so to speak. So we can give our drug, and particularly in that combination setting with Ibrance, we can get the tumor to start disappearing. It actually sort of melts away within about a week, and within about 15 to 20 days, the tumor's completely gone. And in that experimental setting, we then stopped treating the animals. The tumor was gone, and we said, well, let's see what happens. And the tumor stays gone, stays in that, minimally residual state for over 40 days. And so in that sense, perhaps we could think about melting tumors away and not only increasing lifespan, but really causing the tumor to stop growing and disappear. We'll have to wait and see, of course, when we get to humans. And as I always say, that humans are not just big mice. We're a lot more complex. But, I think that the concept of converting cancer into a manageable disease is really our goal, is so that if you could shrink a tumor significantly and then keep a patient on maintenance therapy to keep that tumor in that really small space, that is a real win, right? Our patients will be able to continue on with their lives and, have productive increased lifespan, but also really good quality of life. And so we have a lot of clinicians that work on our particular scientific advisory board, 3 very famous clinicians who sort of run clinical trials at the Dana-Farber, Memorial Sloan Kettering Cancer Center, and Columbia University. And they've really kept us very patient-centric from the beginning. Not only, strong biology, but what's the patient journey? What do these patients want out of this therapy? And it really is, of course, more days, but also really good quality of life. So we're really thinking about that toxicity, that high specificity. That's first and foremost in our mind all the time.
When the cancer that you are able to break down, what happens to the cancer? Where do the component products of that tumor go?
Yeah, that's a great question. So our therapy called IPY induces something called necrotopus. And necrotopus is a big fancy word for programmed necrosis. And you may know what necrosis is. It's when a cell dies and it kind of atrophies and it kind of just stops living. And so our therapy, IPY, induces that form of cell death. And necrotopus, actually, imagine it's like the cell is a balloon, and necrotopus forms something called the necrosome. Imagine it's a little portal on the size of the balloon, and it causes the balloon to pop. And so that's what happens to our cancer cells. They sort of pop, and then all the contents of the cell spill out. And we do think that is stimulating the innate and the adaptive immune systems, which are the systems that our body has to protect us both from external pathogens, both known and unknown. And that's a really good thing. You eventually would like the immune system to be stimulated by your treatment, because then you could harness the body's own, army to eradicate any cancer that is still remaining. So we do think that we're causing the tumor to undergo this necrotopus. It melts away, it pops, and then it gets sort of chewed up and eaten up by the immune system. And that causes a further response to help keep that tumor at bay. So clearly, a lot of that biology still has to be worked out, but that's what we're seeing in our early studies. And we're very excited about that because, you need that cancer to disappear. And we'd really like to stimulate the host immune system along the way to get that to kick into gear and do its job and help eliminate disease even more.
You gave a presentation recently at BioFuture. What was the basis of that?
Yeah, so BioFuture was a conference that was held here in New York City, where we're based. And it was a really amazing conference because it brought together leaders in the field from all areas of drug development. So scientists, Leroy Hood was there. He's a real leader in the molecular cell biology field, but also venture capitalists who have funded lots of early stage work and really helped push that work all the way through to the clinic. Lots of biotech CEOs, CEOs, lots of biotech chief scientific officers, and then lots of manufacturers and lots of contract research organizations that assist in the testing of drugs, the development of drugs, the manufacturing of drugs. So it was all these people were coming together. And it was really, first of all, it was amazing to be back in person because I don't think I'd been in a real scientific conference like that in a while. And everyone was excited to be there. So people were just talking in the hallway and listening to presentations. And we were really fortunate, and we gave a presentation about Concarlo and our novel technology, P27 targeting. And it was an opportunity for us to showcase what we've done, what we plan to do, how we plan to get to the clinic, and an opportunity to interface with both biotech greats as well as, the venture capitalists. We are, in fact, raising Series A funds to push the manufacturing and the development of our product towards the clinic and to also pay for a clinical trial. So it was a really great experience to showcase the Concarlo technology, all the amazing work that my team has done over the past few years.
And where are you at currently, and what's the timeline going forward?
So we are, as I mentioned, we're about 15 to 18 months from having what's called the investigational new drug application, which is the piece of paper that the FDA gives you to test your drug in humans. And you have to put a big package together and show that the drug is safe and to show that this is why you think the drug is going to beat the standard of care medication. So we are finishing up our IND enabling studies, testing the drug in different animal species, looking at how long it lasts in the blood so we can extrapolate what would be our human dose. And we will present that information and hopefully be granted that IND and then start dosing our first patient in 18 to 24 months. We intend to bring in several tumor types into our first clinical trial. It will be, of course, mostly a toxicity trial showing that it's not harmful to humans. But in the oncology space, we actually test our drugs on patients that have disease. And so you do frequently get early readouts of efficacy, and we'll design the trial also to include what are called surrogate markers to be able to tell, did our drug hit the target? Did it do what we thought it was going to do, even in that early stage? So we'll have what are called expansion cohorts. We'll pull out some of the groups and keep dosing them to see if we can actually see those good effects, drive tumors into remission and drive tumors into that MRD, minimally residual state. So very exciting, raising money to finish that. Drug development is expensive, and you need not only the resources, but you need the partners and, the additional expertise. So we're getting out there and we're talking lots to people to present the P27 targeting story, to present Concarlo, and looking for colleagues and friends and partners and investors along the way.
Obviously, what you're doing in itself is great, but is there anything else in the pipeline?
We do. We believe in the power of P27 targeting. This is the natural inhibitor of the three highest value targets in oncology right now, right? Everyone wants to drug these targets. So we think we have a unique way to do it. So we do actually have other modalities to inhibit P27 in our pipeline. So in addition to our biologic approach, which is closest to the clinic, we do have designed some small molecules. And so small molecules are actually little tiny bits of chemistry. Think an aspirin, that's a small molecule. And the benefit of those is they can be given orally, so patients can do that at their own home. But we developed a series of assays, in fact, 5 of them that work in tandem, and we interrogated big chemistry libraries, and we found some small molecules that look like they have that same functionality that our peptide does. So we're in the process of developing those into bona fide drugs, so increasing their permeability, increasing their affinity for the target, and we will be doing that for about another year and a half, and then we'll have what's called our developmental candidate, and then we'll push that forward to the clinic. So that's about 24 to 36 months behind. The two approaches are not really competitory. They're really giving us more shots on goal and the ability to really think about different tumor types that are more amenable to a small molecule and tumor types that would be more amenable to the liposome formulation. So really thinking about the power of P27 targeting across the whole oncology space and trying to think of ways to use P27 targeting into lots of different tumor types. So very excited to have this secondary asset in our pipeline and our secondary approach, and eager to see what happens as we push both of these forward, because then that opens up the world to even deliver these P27 targeting via additional approaches. And we have intentions and desires to move into additional targeting spaces as well.
Plenty to keep you going over the next few years then.
money to keep us going. Yes. And, I'm really grateful to our amazing team of scientists and clinicians and, all of them that just, I'm humbled and in awe of the people that I work with every day that bring just such character and rigor and scientific acumen to solve this problem. And also really, I think people work in small biotech because they believe in the vision and they believe in the power of science to change people's lives. And so that's been something that I've carried with me since my earliest days in graduate school. I worked for a very famous man, and we worked in the early 90s on HIV and retroviruses. And a lot of what we did in that space really helped set the stage for the reverse transcriptase drugs and the integrase drugs. And so I learned really early on that what you do in the lab can translate very quickly into changing patient experience. And so I'm honored to work with the amazing people that believe that in that same vision.
That's great. Is there anything else that you wanted to talk about that we hadn't covered?
I, of course, love talking about Concarlo. I love talking about P27 targeting. For me, this is more than just a job. I actually worked at Sloan Kettering in the mid-90s, and the protein P27 had just been discovered, had just been identified, and I was essentially the first postdoc into that lab working on it. And I've continued working on P27 for the rest of my career. So over 25 years now, I've been studying this one protein that interacts with these three high-value targets. And so early on, we started thinking about, could we use P27 as a modality to inhibit these kinases? So for me, this is an amazing evolution of my personal journey from, researcher to I'm a professor at one of the state universities of New York to now, being at this biotech with the idea of, actually dosing our first patients in just a little under 2 years. So this is a really important journey for me and really the culmination of what I've been doing for a really long time.
And that's it for another week. If you're a football or soccer fan, I'm sure you're glued to the screen for all the games. Sadly, I will miss about a week of them by being on the road, but that's okay. It's always nice to be travelling and going to events. So it's time to go and pack again. And as I'm going through Germany, that involves face masks. Although no need to worry about where my raincoat is because I wear it every day. Although I should probably find some warm clothing. Actually, my bag is almost full already with my camera, so I hope I don't buy anything at the Christmas market. So I hope you enjoyed this week's podcast and that wherever in the world you may be, you have a great week ahead and join us next time for another Beyond Biotech.